Bismillahirrahmanirrahim. Our today's topic is downstream processing. So you all know that in industrial biotechnology, downstream processing is a very important step in order to purify the industrial products. So first of all, I would like to define the downstream processing. Downstream processing refers to the recovery and purification of biosynthetic products from natural sources as well as animal and plant tissues or fermentation broth. So it means that downstream processing has two components. Either we are purifying our bioproducts from the fermentation broth or an artificial media, or if we are also purifying our desired products from the natural sources like animal or plant tissues, this is also a downstream processing, right? And then which biosynthetic products we could purify by using the downstream processing? There are many very important industrial products that could be produced, that could be purified by using this process. Those products include pharmaceuticals, antibiotics, hormones like insulin or human growth hormones, antibodies, vaccines, enzymes, natural flavors. So these are uh, most widely produced biosynthetic products that are produced in industrial biotechnology. And other than these, there are some other products that could be produced in industrial biotechnology and could be purified by using the downstream downstream processing process. And then there are two terms that could be differentiated, and these terms are number one, downstream processing, and the second is analytical bioseparation. So what is the difference between downstream processing and analytical bioseparation? There is a very minute difference. So downstream processing refers to the separation of bioproducts in marketable quantities. So it means that when we produce our desired bioproducts in very huge quantities and we package those bioproducts in order to market them, then it is called as downstream processing. And in the second term, that is analytical bioseparation, that implies the separation of bioproducts for for the measurement of components or components in a mixture. So it means that when our purpose or our aim is only to measure any single component or components in a mixture in a laboratory, or our aim is only to measure those components, then it's a analytical bioseparation. So this is a minute difference between downstream processing and analytical bioseparation. Downstream processing plans goes in the markets. So in downstream processing, we produce our desired bioproducts in huge quantities in order to market them. While in case of analytical bioseparation, our purpose is only to analyze, our purpose is only to measure the single component or many components that are present in a mixture. Right now, I'm going towards the uh, steps that are involved in downstream processing. So there are some steps that are involved in downstream processing. Number one is the removal of insolubles. Number two, product isolation. Number three, product purification. And number four, product polishing. So now we are going to discuss one by one all these steps of downstream processing. So the first step of downstream processing is called as removal of insolubles. What is removal of insolubles? It is the separation of cells, cell debris, or particulate matter from the fermentation broth. So removal of insoluble is actually composed of two parts. One, when we remove these insolubles from the artificial media or fermentation broth, and in second type, when we remove these insolubles from the animal, from the homogenized mixture of the animal and plant tissues. So in the first type of the removal of the insolubles, we separate out the cells, cell debris, particulate matter from the fermentation broth. And it means that when we are producing certain bioproducts in industrial biotechnology, so after the fermentation process, the very first thing that we do is we start separating out the cells, cell debris, and particulate media, particulate matter that is present in the fermentation broth from the supernatant. So it means that we separate out the supernatant from these cells, and supernatant is actually the media which contains our desired bioproduct. So some techniques that could be used in the removal of insolubles from the fermentation broth are centrifugation. It means we can centrifuge and separate the supernatant from these cell debris. We can also filter out. We could use some filters in order to separate out this debris, and in order to pass through the liquid carrying our desired bioproducts. And sedimentation is another step. Sedimentation could be used, and we sediment all these cells and debris and particulate matter at the bottom, and separate out the the liquid containing the desired products. And we could also precipitate. We could use the process of precipitation. So precipitation is the is to separate out these debris by forming their crystals. And flocculation could also be used. So flocculation is forming a bunch, forming a clump of the solid materials in the fermentation broth, and then removing it out from the fermentation broth. This type of step is called as flocculation. Then in the second type of the removal of insolubles, we use what we do is we recover our product from the animals and tissues. And this type of removal of insolubles also involves some steps, like we discussed the steps in case of the fermentation broth. So in case of the natural tissues of the animals and plant tissues, the steps involved are grinding of the tissues, then homogenization of the tissues, and then leaching out the desired products from these tissues. So this is how we remove or this is how we separate out our desired bioproducts, and we in the first step, that is removal of the insolubles. Now we are going to the second step of the downstream processing. The second step is named as the product isolation. So it means that now we have removed the solid material. Now we have removed the cells or the debris or the particulate matter from the mixture, and now we are going to remove our product. So for the removal of our desired product from the liquid, what we need to do is we have to remove the water, because water is a molecule that could change the physical and chemical properties of the desired product. So our first step, or after the removal of the debris, our first aim is to remove the water from the mixture. So for the removal of the water, what we do is we again perform some steps, and those steps could be: number one, solvent extraction. So it means that we could use some solvents in order to extract our desired products. And what solvents could be used? For example, we can use ethanol, methanol, and so many other organic solvents could be used in order to specifically extract our desired bioproducts. And the second step is the adsorption. This means that we could adsorb our desired solutes on some solid materials, right? And another step another technique that we can use for the product isolation is the ultrafiltration. So ultrafiltration means we could use such filters, such sophisticated filters that would separate out our desired, uh, that, that would separate out the mixture containing our desired product from the rest of the liquid. Then precipitation could also be used. We can precipitate our desired product or the mixture that containing our desired product in a liquid. So after that, in downstream processing, the third step that we are going to discuss is product purification. So it means that now we have, in the first step, we have removed the cells, we have removed the debris, we have removed the particulate matter from the mixture. And in the second step, we have also removed the water. And the purpose of removal of the water is just to protect our desired bioproducts from changing their nature, right? And in the third step, that is a very important step in downstream processing, and that is called as product purification. These steps involve very sophisticated methods, very sophisticated techniques that are involved in the separation of those components that resemble the product in terms of physical and chemical properties. So it means that now we have a mixture that is free of the cells, that is free of the debris, that is free of the any particulate matter, and that is also almost free of the water contents. Although it contains some water, it might contain some water content at this step, but it is free of the water. So it means that now the mixture contains only might contain some of the solutes, and the solutes may resemble in terms of their physical and chemical properties. So here our purpose is to separate out our desired product from many other solutes that may resemble in terms of physical and chemical properties with our product. And these steps, like the previous steps, also involve some techniques, some steps. So what methods this step involve is, uh, this step of product purification involve very sophisticated techniques like affinity chromatography. So what is affinity chromatography? Affinity, affinity chromatography means it's a chromatographic technique in which we separate out our desired molecules on the basis of the interaction of the like antigen antibody reaction, like substrate enzyme substrate reaction, like receptor ligand reaction. So when we separate out our desired product in chromatographic technique on the basis of their interactions, then this type of chromatography would be called as affinity chromatography. Then the second type of chromatographic technique that is used for the product purification is called as size exclusion chromatography. So from the name size exclusion chromatography, from the name it is obvious that it separates out the biomolecule on the basis of their size, and in some cases on the basis of their weight as well. So when we separate the biomolecules in chromatographic technique on the basis of their size, and in some cases on the basis of their weight as well, then this type of chromatographic technique would be called as size exclusion chromatography. Then another type of chromatographic technique is used for the product purification in downstream processing. This is called as reverse phase chromatography. So reverse phase chromatography also separate the solutes on the basis of their hydrophobic interaction. So this chromatographic technique separate out the molecules on the basis of their hydrophobic interactions, like hydrophobic interactions of the solutes that are present in mobile phase, and with the interactions of the ligands that are coated with the stationary phase. So these type of the separation takes place on the basis of these types of interactions. Then another type of technique that is used for product purification is called as ion exchange chromatography. So ion exchange chromatography uh, is obvious from the name that it separates the molecules on the basis of their charge. So here our purpose is to separate out the polar molecules that have the charge. So we separate the molecules on the basis of their charge. Another type of step could be involved in the product purification, that is called as crystallization. So it means that we can form the crystals of our desired bioproduct, and we can separate the desired product from the other impurities or other solutes having similar physical and chemical properties. And finally, we can also use fractional precipitation. So in fractional precipitation, we can precipitate out our desired product from the other solutes. So this is how we use some of these very sophisticated techniques in order to separate our desired bioproducts from many other molecules that may resemble with our desired product in terms of the physical and chemical properties. So finally, in the last step of the downstream processing, the last step is product polishing. So once we have gone through all the steps of the debris removal, of the cells, and then removing the water, and then extracting our desired product, and then separating our desired product from the other similar products. Finally, our final aim is to polish the product. So in this step, what we can do is we can finally dry our product, we can sterilize our product, and we can package our product. So these are some of the steps that are involved in the final polishing of the product. And this step also involves some methods, some techniques. For example, desiccation. So what is desiccation? Desiccation is the removal of water, removal of the liquid from the desired product. So in desiccation, what we do is we create a vacuum, and we remove the water contents by creating by a specialized instrument that is called as desiccator. Then another technique that is involved in the product polishing is called as lyophilization. And I think you might have well aware of the process of lyophilization. Lyophilization is actually the freeze drying. So freeze drying, and there is a specialized instrument that is called as lyophilizer or freeze dryer that is used for lyophilization process. So here, what we do is we remove the water content at extremely low temperature. Then we can also use a spray drying process, and for sterilization, we can remove viruses, removal of viruses. Then finally, we can also add a step of depyrogenation. So depyrogenation is also a sterilization step in which we remove the pyrogens from our desired product, and pyrogens are toxins that would be produced by the bacterium during the fermentation process. So this is all about downstream processing and its definition, its importance, and steps that are involved in downstream processing plans. So this is all about today's lecture. If you have some questions, you can ask.